Hi, everybody. This is the recording for this day of absence or unavailability, September 12th. And I'm recording to you, I'm going to send everybody the one on the Biol 1210A. So don't be surprised if you see that identifier when I embed that as a YouTube, but it's for everybody. And because we're at slightly different places, I was going to take this in a slightly different direction because it's material we're going to start to cover, and it's background material. So I do not have this uploaded yet onto Blackboard, but where I am, it's difficult to get a signal. So I'm not even sure where this will be sent, when this will be sent. Uh, Certainly before we return on Monday, it'll be sent, hopefully in enough time that you can listen to it. But what I'm going to be doing, and I'll show you the PowerPoint here, is using this because I can introduce the cell in most of the classes, effectively all the classes, I have to explain the structure of proteins and DNA and RNA. It's very difficult to do just narrating and looking at PowerPoints. I need to be able to demonstrate it a little bit differently and do stuff on the board. And this doesn't lend itself to it. So rather than being into that lecture 2 or whatever we were going to do, that would have been in 2B. This is from the same textbook, the Marie textbook. This is their introduction to the cell. And again, I like to use these because of the animations and then examine both the Walsh Hull textbook, which is the textbook we utilize for your class, and which has all the same information. It's just the way it's presented is a little bit different. So hopefully you're able to hear me. And I hope it comes through again, because I may lose this at times. So it may have to come in sections. It just depends on how well the internet, the Wi-Fi they have here. I don't even have a phone signal where I am. So that's the way it is. And so this is what, and I will load this for you. Hopefully, surely Sunday sometime would be the latest it would be because I'll be back home by then. Nevertheless, we're made out of cells. I'm going to show you a little protein animation just when I get to one part about this. And again, this will serve because it's really, and I'll even review that down the road because I think it's so darn important. But this will be the first look. So it used to be if you understood the cell by itself, you really had no idea how things worked. Okay, these are bone cells, these are white blood cells, whatever. Now, to understand the cell, we have to look inside of the cell. And understand the molecular biology of it to a certain extent. But at least this will give you an idea of where we start this basis and give you an idea of the complexity of the human cell. It's remarkable. And again, this is the distinction between prokaryotic, which are cells like bacteria, which don't have a membrane bound organelles, versus our cells, eukaryotic cells, which do. And the distinction is not just based on this. If you take lower order animals that are still considered animals and or lower order eukaryotic organisms, they don't have the complexity or the interworkings. I mean, similar, but it's not the same. It's effectively our cell is as advanced as it can be, and particularly when you get to muscle and nerve cells. So, good old cell theory. When you use the term theory in biology or in any science, a theory it's like the theory of relativity. I mean, we call it accepted science. It's almost no one has been around to disprove it, though people have tried with all of these theories. So we accept this. It's just when we finally get to the point we say, well, there's no way this could possibly be wrong. Then we get a law, like Newton's like laws of gravity. Okay, a cell is the structural and functional unit of life. We're all made out of cells. That's what it is. The entire organism depends on its cells. Structure and function are complementary. We deal with this every time we have a lecture. Biochemical functions of cells are dictated by the shape of the cell, subcellular structures. They're all elegantly designed. When it has lots and lots of tentacles and surface area, it's going to be sampling. It's a white blood cell. It's on defense. It can grab a bunch of things all at once and begin to process them, which is you'll see with bone and surrounded by a network of proteins that have calcium laden with them and makes them stiff, so it's strong. And so if we were all made out of cells, and all living things are made out of cells, and moreover, they all come from pre-existing cells that are there. And so there's an immense amount of diversity, which you're just starting to see. I know you've been looking now with Dr. Robbins in the lab and have seen the unit on tissues and the unit on skin and starting the unit or maybe into the unit on bone by this time. But we're going to deal with it on perhaps, I'm sure, a different basis than all of those things. I'm not going to reiterate. I'm going to talk about skin later in our second unit and just get back to reinforce the importance of skin, though I don't think it's going to be a major component of examinations, but the cell certainly is. That's there. And all those tissue types, I talk about them all the time. Down the road, you'll hear it a zillion times. I'm a nut about epithelial tissue. So get used to hearing that because it's so darn important. So all of these things, and this is just a standard look-see at what cells are like. I mean, they're all different kinds. So you have here red blood cells. They're not even a cell. We call them in the bloodstream formed elements. Why? It's not a complete cell in the sense there's no, if you look, it's no nucleus. Again, and I use the term all the time, elegantly designed. What has happened as it has matured? Okay, it sheds its nucleus, it sheds its organelles. It's just a uniquely structured container for hemoglobin. And that hemoglobin, and you'll see that in the animation, is something the way it's shaped fits in an immense amount in a tiny red blood cell, as you will see. Immense number of molecules in there, all about being shaped for carrying oxygen. So that's where we marry the shape. This is all about surface area by creating this sort of like double beveled things. So we fancy names called biconcave disc. It's concave here. If you flipped it over, it looks the same. And all that means it's a lot of surface area rather than being like just like a little flat disc that you know what I mean, like a checker for a checkers game. Here, you already looked at fibroblasts. Again, long tentacles. They can attach to these fibers. They secrete or give off the fibers. They attach to those fibers, but you can see the cells are in some way, shape, or form attached to one another, but not next to each other. That's what makes them connective tissue versus epithelial tissue, okay, where they're all attached to, they're in a layer. They're in a sheet for the most part. And with subtle variations, some thicker, some thinner, some different shape, that would be a simple cuboidal layer. You can see by the round nuclei that are there. And then we get to these, and this is a classic oddball. So when we're looking at skeletal muscle, effectively, and here's smooth muscle, these are entirely different. Smooth muscle has individual cells that are networked together. Skeletal muscle is unique. We call it multicellular. Rather than having just one nucleus per cell, which effectively all, if not it isn't all, but nearly all smooth muscles do. Here you have this unique system of a highly developed cell that has tiny identical components, and that's where you see these little lines. We're going to go into and discuss those components when we get into our part, showing you how a muscle actually contracts. Fat cell storage. You did adipose tissue in tissues, it's storage. All it is a cell that's laden with fat. The nucleus is pushed to the side. What's interesting about that, the more you have of those in an organ, say the liver, in someone who has consumed alcohol in excess for 10 or more years, now you begin to see significant deterioration from that. It creates an inflammatory component. Okay, we call it alcoholic hepatitis, and then it leads to something called cirrhosis, and the liver no longer functions, and those people have, at best, a very limited lifespan, and at worst, die, unfortunately, of complications without major treatment that's involved. And here's the aforementioned white blood cell, all those tentacle-like projections. Effectively, they can surround particles that have to be gobbled up and adapt, as we will learn how to do it. Here's a nerve cell. So the nerve cell, very long. I mean, it doesn't depict it here. You could have a nerve cell. Think of a 6 foot tall person. Think of a 5 foot tall person. These you could have, and we'll do it in feet, but actually it's in meters. You could have a 1 meter, that's more than a yard, a 1 meter long individual cell. That tail, called an axon, could be a meter long. It's a 2 nerve circuit. If I want to wiggle my big toe, there's 2 nerves, one from my brain to, the lumbar sacral area or low back area and of the spinal cord, and one from the spinal cord out to your big toe, or just about to it. How's that? Pretty darn long nerve, two nerve circuit. And here you can see something that's really not, again, not a true cell. In a sense, it is, but it's sort of unique. It's just genetic material, okay, that's there. So it's really not a cell in the sense, you can call it a sperm cell if you will, but it's got half the genetic material. It has no organelles. It's just a unit of genetic material. As you can, you don't learn this now, but you learn this in AP2. It's got a head that contains chemicals just to penetrate an egg, and basically a motor here and a tail here, just so it can swim. So it's basically an entity, for lack of a better term. Okay, so they'll talk about sperm cells, and I mean, that's a fine point, but I'm just trying to give you an idea that just like a red blood cell, which is not really a cell, we call it a formed element, this would probably be almost in that category, but we will say sperm cell. Some people do. This is a true cell. That's a white blood cell because it has a nucleus. And you'll see platelets are just little fragments of a cell that have protein granules in them. So all cells have some kind of structure and function that are similar, structures and functions that are the same. The main ones are this: the plasma membrane, and that's the beauty. It's not just flexible. Okay, it's not flexible. Something that you could take a balloon and say it was somewhat flexible in that regard. But imagine a balloon that you could open and close. You could open it in the middle and push it back together, and it wouldn't lose the air, and it wouldn't lose this, and it wouldn't lose anything that was contained within it that were there. So it's flexible, and the cells are remarkable in the sense that when a white blood cell leaves your bloodstream, your blood vessels have tiny openings, and the white blood cells with the aid of certain chemicals, is able to actually almost slide through a little opening. Like if somebody parked next to you in a parking lot was so close, you had to turn sideways and try to get through. And sometimes you can't because some dummies parked, blocks you from getting out, and you're going, oh, I can't get in my car, get out of my car, that kind of stuff. But white blood cells can do that. They can actually change their shape with a degree of flexibility. Just for an example, and it's not the only example. The cytoplasm, very, very similar in its chemical components, but a lot of times there's other stuff in there. Those aforementioned muscle cells, skeletal muscle cells that allow us to move, they're filled with protein rods everywhere. There's remarkably little cytoplasm. We give it a special name you will hear about called sarcoplasm, SARCO. That's a prefix for this when we get to the microscopic level, okay, and the submicroscopic level of skeletal muscles. But there's very little compared to cytoplasm that's there. It's just filled with protein rods. It's nearly all protein. And the nucleus, DNA is the control center. It is also not the only area, if you did not know, where there's DNA in a typical cell. Where do we get to mitochondria? They have their own DNA, and it's very interesting. It's something you deal with a little bit more in micro, but it's an important thing to understand vis-a-vis DNA. And I'll talk about it when we get to the organelles that are there. So that's one of those, the only place in your cell that has DNA is the nucleus. Bang, false. Okay. Again, when you hear only or all or always or never or it's never anywhere else in the cell, bang, false. Voila. You see this diagram everywhere. What it tells you, it's not by size. This is just, you're using a generic cell. No, the nucleus does not overwhelmingly take all the mass of the cell, etc., like this. There's not always this amount of what happens to be called endoplasmic reticulum. We have something that's called rough, which is closer here. We have something that's smooth, and there's not always a ton of rough and very little smooth. And indeed, as you will see going forward, when we have more of one than the other, it tells us function. Structure and function are related. So the nucleus isn't always this big. They all have this thing called a nucleolus. And this is, everybody, always a test question. Okay, always. That means it's on the test. Pay attention. The nucleolus or nucleolus, however you want to pronounce it, is where is the general assumption that's made, and it hasn't been disproven, is that ribosomal RNA, which we'll get to when we talk about RNA and DNA, is made there. Okay. And yeah, it's got also, we'll talk about what chromatin is. We'll talk about these nuclear pores and what they do, and the fact it's not a single membrane, it's a double membrane. You can see the double layers that are there. All of that is relevant. Okay. And again, the amount of this endoplasmic reticulum, you'll learn that when you have a bunch of this stuff that's rough, it's making a lot of proteins. When you have a bunch of stuff that's smooth, it's processing, particularly processing fats. You'll learn about this. Here's the mitochondria. Again, and cells that are busy have a lot of them. Cells that are not don't have a lot of them. Golgi apparatus, we'll talk about its role with regard to all of these membranes and what it does with some of the proteins that are made and how things are spit out, and by the same token, how things are brought in. So we'll look at all of these structures without going crazy about it right now. We're going to look at all of them. There's a whole section, and you probably have had a lot of this stuff before, and that's fine. I'm going to try to explain to you the interrelationship and what are the really what the important factors are about all of them. They're all important, certainly, but really what when it comes to their job description, so to speak, in inside of the cell, what it actually means and what it actually does. So that's we always see that, and the beauty of that. Let me just get back to it. Is as you can see from the other cells that we saw, this looks nothing like those necessarily. This is a fairly simplistic cell, if you want to take it this way. Very generic would be a better word. And so we have things that are inside the cell, intra, intra, means inside, extra, outside. If it's between two cells, we call it inter. Okay, so you hear about fluid in between cells, or in between tissues, interstitial, interstitial, interstitial between tissues, versus extracellular and intracellular. And we're going to talk about their fluid composition, as you can see here. They're very, very similar, but not identical. And they have a lot of, they may have different chemicals, but the balance between them we will get to. So the interstitial fluid, the plasma, cerebrospinal fluid, all fluids, saliva, mucus. Mucus is still basically fluid, even though it has more protein, which makes it thicker and kind of stickier and yuckier. And all of this, and their connective tissues. You talked about the matrix, this combination of ground substance and fibers that are embedded together. And what they do is effectively, it's a good way to describe it, intracellular glue that holds things together. And if you want to go where the glue thing comes in, how I do not know how they make it today, but they used to effectively, the collagen was something that was rendered down to make glue. Okay, over the years. So really, originally our glues and paste, not so much paste, but these are the glues that we use came from animals, and basically they were after they died, they basically cooked them down and rendered or extracted what became glue from them. Yuck. Okay. And so the membrane itself, and that's really just a little focal point for today. I'm not going to get into movement. I want to just show you what it contains and we'll work from there. So it acts as an active barrier, really, of separation between these two structures, between the fluid on the outside and the fluid on the inside. And you can see the fluids are similar but different, and it's actually going to control the movement of particles between them. So basically, and it's one of the points of emphasis that I will tell you that we will have on this first exam is called movement across membranes. It's as important as anything that I will tell you about this whole semester. Every membrane is a semi-permeable membrane. In short, some stuff can get in and some stuff can't. With that semi-permeability is what sets up the remarkable ability of cells to do different things. Why we have muscles that move, why we have nerves that conduct impulses, why other cells do not, is all about the ability to move across membranes. The cells are allowed to do business as long as they have this relationship when they can move basically particles in, out, to do energy transformations and out to get rid of waste products. just to start with something basic like this. And yet we call it the cell membrane, plasma membrane, cytoplasmic membrane, all terms created equal. And this is what we got to in each of the classes, the idea of the phospholipid bilayer. That is the foundation for all of these. The fact that every one of those phospholipids naturally form that bilayer when they're put into water is a remarkable function and enables the membranes to form. And then they have the ability, as I commented on, and we'll look a little bit about it today with this animation and some of the illustrations that you will see here, why proteins are proteins, why they can do this stuff, why they have different roles within the membrane, and how they allow different things. And you're going to see 6 of them in this particular start of the PowerPoint, which is also an important part for the test. So they can float, okay, different patterns. So we use the term fluid and mosaic model. The mosaic, because the proteins are all different and they can move around to some extent, an interesting pattern. The fluidity enables their movement, and the fluidity enables the flexible and shape changes, and the fluidity also allows it to open and close. And you're going to see every single cell is covered with sugar, small sugars. I commented on it when we talked about phospholipids. Little sugars protruding are an identity feature. Okay? It's what genetically makes our blood type. Okay. Collectively, we talk all of those sugars, and not just sugars. They can be sugars that are attached to fats, sugars that are attached to proteins, all sorts of things. We call that a sugar shell, hence the term glycocalyx. Glyco for sugar, calyx for shell. And for instance, you're going to see some of these unique aspects of proteins allow the cells to be held together, like you saw in epithelial cells, okay, or attached to fibers, like you saw in connective tissue. Cells rarely exist in a vacuum, maybe with the exception of white cells. Red cells work aggregately, but they're not attached. White blood cells are definitely not attached to each other. They may attack in unison to a target and try to surround it, but again, it's more like an army of multiple individuals. And that might be a good way to express it. And here you can see it's not, again, I don't like illustrations where they're busy. But here you can see the phospholipids in the heads and the tails. Here, we're going to talk about it because not every class did I get a chance to talk about cholesterol, particularly in the A section. But cholesterol, as you can see, embedded in here accounts for the stability. Typically, when I finish the cell unit or even into the cell unit, well, maybe halfway through, when I start to get an idea of the structure, there's an animation that's been around from Harvard for 25 years. And you probably have seen it. Okay. A lot of the students see it because it's made the rounds all the time, but maybe even a little dated by this time as we understand more stuff about it. So here you can see all of those things. Decreases water solubility of the membrane, the cholesterol, so water can't get through it. It can stiffen it, but it also, a lot of times, these will serve as platforms for the proteins to do business. This is what cholesterol looks like. We'll go out in the class that didn't have it. We'll go over it again, but it's a very distinctive structure. And you can see these molecules are everywhere. And again, this is not to inside. These proteins are enormous compared to the membrane. The membrane is tiny. The proteins are huge. They can't draw at this scale and really get a lot of it. So here you can see phospholipids, cholesterol, different anchors, okay? Lipids that anchor or structure to the proteins, because understand that proteins have a lot of lipid compatibility. It depends, as we will see when we get to the amino acids and proteins. And that's why it's a little bit of cart before horse, that proteins have the ability to be water soluble in certain areas and fat soluble. So they can tether into there. And they're going to talk about proteins that are integral and really part of the membrane, and proteins that are peripheral. Okay. And peripheral proteins protrude. And it isn't just outside, they protrude outside and inside. And oh, that's a big deal because their ability to be something can attach to a protein on the outside and impact what's going on the inside because it causes it to change shape. And here you can see, now they didn't draw it on all of these, but every single protein is going to have not just adenosine molecules, which carbohydrates, but they're part of, again, white blood cells. This is getting back to the blood types and things, whether it's friend or foe, if you want to consider it that. Okay. But there also is a certain identity feature that's there. Okay. It tells us what cells, when we can look at those features and look at the sugars that are on them and know specifically the role that it has, are found only on the outer surface of the membrane, like the sugar coating on a breakfast cereal. And that's where the term glycocalyx comes from. Very small change. You can see there are little hexoses. You can see hexagonal shape. They're short chains. They're not huge. Okay, but they're enough to make the identity. I mean, here the scale is probably fairly accurate. It's there. And here you get the idea of what it does. The fact it's a physical barrier, okay, enclosing the cell, the selective permeability, the ability for cells to communicate with one another. Cells give off chemicals, and in certain areas where the cells are tethered together, they have little protein passageways that send either electrical or chemical signals or both from one cell to the other. When you get to bone, you'll see that's how they work. Okay. And again, we'll talk about recognition. So there's a lot of activities that are associated here with the plasma membrane. And I wanted to underscore a few and take a look at some of the proteins. The proteins that you're looking here, okay, are called globular proteins. A glob of proteins. Well, a glob of something down, some type of stuff my grandchildren play with. And we're going to go back real quick. They are different than the collagen or the elastin or the reticulin that you learned about in connective tissues. Those are fibrous proteins. Their construction is a little bit different. That's something I'll do for you in lecture when we sort of step back and take a look at this. I just wanted to point out that distinction that's there. They have some of these. I don't know that they're very good. I'll listen. Must be bad. Try again. This is probably my internet connection. We'll skip it. That's what I meant. I have a limited connection. I know this is probably recording. I hope it is because I'll be embarrassed if it doesn't. Hold on a second. I'll look. Let's take a look and see if it's recording. Please tell me you're recording. Okay, so it is, as far as I can tell, it is recording. So I'll go back and take a look. Sorry to do that, but I hate to have to do this again. So here you can get an idea of what the membrane looks like. 75% phospholipids. And again, hydrophilic heads, hydrophobic tails. And all it means is that the heads are on the outside, as I did in all the classes that make up the membrane, or on the inside, and the tails really comprise the membrane. Glycolipids, lipids with sugar groups, again, on the outside. We even have, lipids that, and they play a role in a variety of things not relevant to us now. And the cholesterol, 20%. There's a fair amount of cholesterol. Again, it's not like you hear, oh, there's so much cholesterol, my cholesterol is so high. Understand that they're sort of mistaken. Oh, I'll never eat an egg again. It's filled with cholesterol. Are you nuts? I mean, people, it's the problem with cholesterols are cholesterols that are made, excessive cholesterol and cholesterol-like substances that are made principally from sugars. Okay. It's not the cholesterol that you're ingesting is the problem. I mean, if you're going to eat an all-egg diet, that might be a mistake, but but excessive anything is proven to be bad. And so here's what happens. Okay, again, the hydrophilic head and the hydrophobic tails. And this would be an example, not so much of a cell, but what it's holding in here. And indeed, you might remember me talking about lipoproteins a lot of times, and we call them, you don't have to know this, very low density lipoproteins. The way if you gobbled up that big bag of good old oily potato chips, that giant thing of fries with everything on it, okay, that basically they would trap all of those fats within here, fats attached to proteins as your blood were trying to go through your bloodstream and surround them. Oh my God. That's what they have proteins on the outside, and they'd have a very low density because it was all fatty and loose. And it's called a mycel. I mean, that's just gives you an idea what it would be like. And this is really the point of today's little talk on what proteins do. They communicate with the outer environment. Half the mass of the membrane. The membrane itself, if you think of the fatty component, is not nearly holds as much weight as or as much mass as do the proteins do. There's a ton of proteins. They're very specialized. As you could see, there are areas where they're tethered or held onto within the membrane, don't move. Okay. And other ones that can move somewhat freely. And so basically integral proteins are just that. They're just confined within the membrane. Whereas peripheral proteins are ones that are attached either to the inside or the outside and protrude into the environment. These are integral, firmly inserted. We call them transmembrane. They go all the way across, okay? And they have areas on the protein, as I was saying, that attract fat and items that attract water, so they can nestle very nicely in there. And typically, these are what we call channel or carrier proteins, as well as enzymes or receptors. And you'll see these in maybe a second or two, a minute or two. And so, and I'll try to explain the difference, and I'll probably have to explain it a little better when we get back to class. Peripheral proteins are attached to integral proteins. And again, filaments are intracellular surface used for plasma membrane support. So think of connective tissue. They're supporting those cells. Those cells aren't designed to move around. Those cells have to have something to hold on to. And there's other ways that they work. An enzyme, okay, as you will see, will create alterations typically in the exterior, and basically on the exterior of the cell will have chemicals that are on the exterior, either be able to make them into something, or better yet, make them apart so we can absorb them. Motor proteins are a whole different area. This is something that people don't understand. It's the inside of the cell is not a static environment. The mitochondria doesn't stay just there. It's constantly being built and rebuilt. The cell alters, it can change shape. Cytoplasm flows throughout the cell. We're constantly making little roadways and passageways made of things called microtubules or microfilaments that allow this to change shape. Okay. And motor proteins are just that. They're proteins that can change shape. It's the way our muscles work. The motor protein called myosin changes shape. And in doing so, it lashes onto another protein called actin, and the actin pulls on a component inside the cell and basically creates the movement that you and I call muscle contracting. And yes, they interact with holding cells together. These are the 6 different ones. I don't want to go into them in detail with this. Okay, I'm not going to show you that slide, but I will slide that and make a comment about each as I get ready to conclude. I'm making this a shorter presentation because I'm not so sure you're going to get it. Transport, stuff in and out. 2 types. Ones that are always open, we call them channels. Think water, think pores. So you're going to sometimes, a name that you'll see associated with them, aqua, water, porins. All one word, AQUAPORINS. They're very tiny channels. Most cells have them. water moves freely, not through every single cell, but most cells have them. What holds the water in or pushes the water out has to do with what's dissolved in the water, either inside and outside. That's why electrolyte balances are so important. When people are dehydrated, okay, or overhydrated, it creates unremarkable electrolyte balances that can be very problematic, if not fatal, that are there. Carriers carry something. They have to change shape. They have to open here or close here or the other way around. And you'll see this in animation. This is not quite what happens, but it's sort of like the opening opens and closes with things that are associated with it. And sometimes to do that, it requires energy. Okay. So what happens is these are chemicals that can't slither through the membrane. So that by definition, a hydrophilic channel, say for a charged particle like sodium or potassium, okay, or something that is hydrophilic by nature and can't get through that's important, like glucose, you will see we have channels that can open and close. And sometimes we have to utilize energy to get them in and out of the cell, and I'll explain why. We have a name for them. In general, we call them pumps. Whenever you see the term pump associated with any of these protein passageways, okay, it's called active transport. Energy is required. So those two terms are synonymous. You hear about, I talked about them a little bit when we talked about pH. Proton pumps that make stomach acid, they require energy. And again, I can't do these as easily because of this. This is the classic example. This is what insulin does. Everybody thinks insulin, oh, and it's like effectively it unlocks the door for glucose to come in. It's not going through the same door. Okay. A chemical messenger like insulin targets the outside of the cell. It causes the protein to change shape and it causes things to occur inside the cell. One of those things that insulin does is it opens passageways like this to allow glucose to come in. So that's what a receptor does. It can be different. It could be human growth hormone. All of our cells have those adapters. Insulin, effectively, all of the cells have those as well. Okay. And what they do is they cause cells to duplicate and replicate and they just have different mechanisms that they accomplish. So receptors are everywhere. We communicate chemically for the most part inside of our body. If you got sick, if you had an infection, whatever you had, and you had to take a pill, okay, a lot of those pills are going to work on very, very specific receptors. They are all the time. And you'll learn that you'll be utilizing these. Many of you go into health science are going to need to know how they work. You're not going to have to know what kind of receptor it is, probably, but you may. Okay, some of them we'll talk about that when we get to the nervous system in a generic fashion. These are enzymes. Think of this as inside the cell. This is outside the cell. And the enzymes are there. And this is really what we see the great abundance of these in digestion. So think of this being able that these are molecules of effectively of lactose. And we're trying to, we have to have that enzyme to break it down into galactose and glucose. That's what they do. We call them brush border enzymes because microscopically, the digestive in your small intestine, they all look like a brush border, kind of fuzzy looking border, like you were looking at a hairbrush. From a distance, you could see the fuzziness, but you couldn't necessarily see the intricate little sort of nylon or whatever material they use for the hairs that are there. And so we'll talk about enzymatic activities that are there. These are the identical, these short chain, we call them glycoproteins. Short chain sugars make up that glycocalyx. And identification tags that are specifically recognized. Effectively, if this was not recognizable, these two cells, and one was a white blood cell, it might attack the other cell. It might latch onto it and begin a process of trying to destroy it, and effectively destroying it for the most part. That's how your immune system's working all the time. It's basically it's not targeting certain; it's just rolling over cells And if it happens to roll over something it's unfamiliar with, it'll latch onto it and big trouble, presume. We'll get more about that as well. And here's what I was talking about before. These, we call the extracellular matrix, that's that same term for connective tissue. Here's the ground substance, here are the fibers. It's got to latch onto the cells, maintains their shape, helps their location of certain proteins. They can move over one another by latching onto something and then unlatching, latching and unlatching. It's very organized. And lastly, we're going to talk about special types, and they got a great name, CAMs, cell adhesion molecules. They love it. Medicine, science, they love acronyms. You got to be careful with acronyms. A lot of times they say the same. They're the same things. And there'll be examples of these we'll talk about. And there's various kinds of those intracellular junctions. And again, I think that's all. I'm going to show you this little animation. Hopefully I won't have to go through the YouTube. Hopefully it'll play. Again, I'm worried about the internet connection. If it doesn't, it doesn't. So I conjured that up over here. Let's see if it plays. What is a protein? I mean, it wasn't rocket science. I'm not using it. They tell me the music is bad. But that's what, those are fiber. You can see the way that these, and I'll pause it. Insulin, antibodies have a different shape. They kind of look like the letter Y. Insulin, big globular protein. Collagen, you can see, you're going to see these proteins. And remember this look, they're braided. It's almost a braided. Structural proteins are typically braided, much more than you would think. So I'm narrating a little bit because they tell me the music with this is horrible. And here they're going to show you, these are called, they're showing you they're called amino acids. You don't have to know the names. We're going to talk about those next time. And that's why I have to draw that on the board. And you'll notice here sulfur plays a role because sulfur imparts a certain degree of structural readiness for a lot of these. And we'll get into the chemistry of it with the carbon, but they all have a similar, and you can see they're using the Tinker Toy models. So they're talking here about hydrophobic, without going into all the names. You're talking about hydrophilic. I don't know what all this is doing. It's not perfect. Here's an acidic. It's got charge. Acids have charge. We'll talk about those. And this is what we're going to really focus on, how they latch together. That's forming a bond called a peptide bond. And together, by latching them together, and that your DNA is directing this sequence, okay, you form, let's say, a linear molecule. We call it the primary structure. And then what happens, you have secondary structures, and hydrogen bonds play the key, and it can form a spiral. We call it an alpha helix. And you can see the hydrogen bonds pointed out in yellow. Or it can form a ribbon-like structure called a beta pleated sheet. Okay, and it's a fold, and folding is very important. I'll get to that. And so when you form a globular, I mean, look what happens. You have a combination of sheets, and it could be charged or uncharged, acidic or alkaline, water soluble, fat soluble, and even within it, rooms to store metals. And when we put that together, now we have something called a quaternary structure. The folding is the tertiary. The quaternary is when actually it increases its function. Quaternary is what most of these globular proteins are. And when you put that iron in there, and look how many, there's 280 billion of those in a red blood cell. And their shape and their functions tell you exactly what I was talking about before: transport, storage, structure, defense, communications, etc. Here's the antibody. Again, it latches on. Let's say it's got a cold. It'll latch on specific points and immobilize it. Structural proteins, different, because generally they're insoluble. Transport. We're going to look at different pumps that allow ions to come in and ions to come out, changing shape. Here's insulin latching onto the exterior of a cell, changing its shape, and things happen. Here are enzymes gobbling up carbohydrates and separating them. and storage molecules. This is how we store iron. Iron is really important to store. So that's everything there. I'm going to stop it here and hope this is all recorded. Appreciate your time. Please give it a listen. I'll embed this and send it along. Thanks again.